Hi everyone, welcome to Dento'ZEN. We are continuing with our topic of morphology and physiology of bacteria. We have already discussed the classification, different microscopes, different staining methods and various shapes of bacteria in our previous videos. You can check those videos. In today's video, we'll start with bacterial anatomy, from which you can get so many short notes for your exam. And in the last, we'll discuss growth and multiplication of bacteria in a separate video. So let's start with today's video. Before starting, quickly subscribe to Dento'ZEN, tap on the like button and hit the bell icon so that you remain notified about new videos. Now, when you look at a bacterial cell, what can you see? You can see an outer layer which is also called cell envelope, which further has two parts. The outermost is called cell wall and inside to that is called the cell membrane or the plasma membrane. So we have cell envelope which has cell wall and plasma membrane. Now inside of this cell wall we have protoplasm, which is made up of cytoplasm and the inclusions, and then we have nuclear body or nucleoid. Now can you see anything else here? Yes, you can see a structure like this here on the outside of the cell. So some bacteria may have some additional structures which are called cell wall appendages, which can be of three types. First is capsule, that is like a layer over the surface of the cell wall. Second is flagella, as you can see here, so they help in the movement of bacteria. Then third one is Fimbriae, which helps in the adhesion of bacteria. So those are appendages. So that is the normal morphology of a bacterial cell. But sometimes we can see atypical forms of bacteria which do not look like a normal cell, and sometimes these bacteria can also form spores. So your long question from this topic can be to describe the morphology of bacteria in detail along with a note on the appendages. So then you have to describe everything. But then you can get so many short notes from this topic, for example on cell wall, on appendages, atypical forms and bacterial spores. So we will today discuss the cell wall, cell membrane, cytoplasmic inclusions, nucleoid, and the remaining topics, that is cell wall appendages, then atypical forms, and spores will take up in separate videos as they can be separate questions. So let's start with the cell wall. So bacterial cell wall is a rigid and tough structure. It comprises 20 to 30% of the dry weight of the cell, and it is just 10 to 25 nanometer in thickness. So can you see this cell wall under light microscope? It is in nanometers. No, we cannot see, not even with light microscope, not with simple stain. Then how do we see it? We can see it by a method which is called plasmolysis. When we place this bacterial cell in a hypertonic solution, its cytoplasm shrinks and its cell wall will remain. So then we can see this cell wall. Other methods are microdissection, reaction with antibodies, as these cell wall have specific antigens which can react with specific antibodies, and then we can see it. Then we can also see it with differential staining methods and electron microscope, as it has better resolution. So these are the methods of demonstration of cell wall. Now what are the functions of this cell wall? First important function, as it is a tough and rigid structure, it provides shape to the bacteria. Very important point. Ductility and rigidity, which is mainly due to presence of peptidoglycans, important component of bacterial cell wall. Another function is protection. It protects against lysis, that is breakdown, and against toxic substances. Also it provides pathogenicity to bacteria, that is disease causing ability of bacteria is because of the presence of so many factors. Virulence factors are there on the cell wall which can damage the host cell. For example, endotoxins are there. So also when we give antibiotics for bacterial infections, so where these antibiotics act on bacterial cell, they act on the cell wall. So that is important, that is site of action of antibiotics is cell wall. Now as bacteria can be of two types, gram positive and gram negative, based on their staining properties, so we should know the cell wall of each type. So let's first talk about gram positive cell wall. Now important component is peptidoglycans, but it is thicker than that in gram negative. About 50 to 100 layers are there. These are also called mucopeptides and they lie in chains. So they are more made up of two molecules, important ones that you have to remember: N-acetylmuramic acid and N-acetyl glucosamine. So these two molecules, they alternate with each other in chains like this. This is NAM, and then in between we have NAG. They are forming chains. These chains are cross-linked through tetrapeptides. Tetra means four. So four molecules, four peptides, as blue color here you can see. Red is pentaglycine bridge between them. So this purple and light blue are the teichoic acids, which are also important components of gram positive bacterial cell wall. So in short, the bacterial cell wall has thicker peptidoglycan layer made up of two important molecules, NAG, NAM, alternating with each other in form of chains, cross-linked through tetrapeptides and pentaglycine bridge. So that is the bacterial cell wall peptidoglycan. So this tetrapeptide may have these four peptides. Then what about teichoic acid? Teichoic acids are polymers of ribitol or glycerol. They are joined by phosphate groups and they maintain the structure of this cell wall, and they are of two types: cell wall teichoic acid and lipoteichoic acid. Now, important point here is that these teichoic acids are absent in gram-negative bacteria. Very, very important point. So, two differences: peptidoglycan is thicker here, and teichoic acid is present here. Now, inside of the cell wall, there is a membrane which is called the plasma membrane, which is like this. So, in between the plasma membrane and the cell wall, we have a space which is called periplasmic space. Now, let's see the gram-negative cell wall. So, peptidoglycan, important difference, it is thinner here. So, only 1 to 2 layers are there, but basic structure is same as mucopeptide chains in gram-positive, made up of two molecules, NAG and NAM, alternating with each other in form of chain, and then tetrapeptide is there. But tetrapeptide here, at its third position, an acid is present, that is diaminopymalic acid, which was not there in gram-positive. And the glycine bridge that was present in the gram-positive is absent here. So that is the difference. So outer to this cell wall peptidoglycan layer, there is a membrane outside, that is here, that you have to remember, that is called outer membrane, which is like this, which is made up of phospholipids, and it is outside the peptidoglycan. So this was peptidoglycan, so this is the outer membrane made up of phospholipids. So this outer membrane acts as a protective barrier, and it has so many proteins inside it. Outer membrane proteins specific are porine proteins. As you can see here, these porine proteins, as the name says, they can cause pores, so they can form channels, diffusion channels, through which the small molecules can pass. Also, they can act as the receptor sites for certain bacteriophages. So they can be the receptor sites, and they can be the diffusion channels. So then another component which is present in gram-negative cell wall is lipopolysaccharides. As the word says, it has lipids and polysaccharides. So let's see the structure. It is made up of three parts. First part is lipid A or endotoxin. Here this is lipid A or endotoxin, which is responsible for the endotoxicity of the bacteria. It can cause necrosis of the host tissue and other effects. Then second component is polysaccharide, that is core polysaccharide. This is the core polysaccharide. Now, as you can see, a side chain here to this polysaccharide, that is called the O side chain, or O antigen, or somatic antigen, formerly called bovine antigen. So that is the lipopolysaccharide structure. So then inside of this peptidoglycan layer, we have the cell membrane or inner membrane. So we can say that in between the inner membrane and the outer membrane, a space is there which is called periplasmic space. It has peptidoglycans. So we already know the two types of cell walls, gram positive and gram negative. Let's see the differences quickly, which can be important short note for you. Let's summarize. So first important is peptidoglycans. They are present in both, but they are thicker in gram positive. Then within peptidoglycan, diaminopymalic acid is present in gram negative, and pentaglycine bridges are present in gram positive. Then second important component, teichoic acid, is also present in gram positive, and it is absent in gram negative. Then what about lipids? Lipids are present in gram negative. So lipids are present in gram negative. Lipopolysaccharides, lipid A endotoxin is seen in gram negative. Then several amino acids are present, and also aromatic and sulfur containing amino acids are present in gram negative. So those are the differences. Now let's talk about the plasma membrane. Plasma membrane or cell membrane is thin layer. It is just about 5 to 10 nanometer, and it is lines the inner surface of the cell wall. So this was cell wall, inside to that is the plasma membrane. So it separates the cell wall from the cytoplasm, and it is essential for the survival of bacteria. Now this plasma membrane is made up of phospholipid bilayer, 2 layers. So 2 layers of phospholipids are there, and this model which describes the cell membrane is called the fluid mosaic model. That is, in this fluid mosaic model, it is a mosaic of so many things. That means this phospholipids are there, then some proteins are there, this is integral proteins are there, and then peripheral proteins are there which are on the periphery, then carbohydrates are there. So this model was given by Singer and Nicholson, and it is the most widely accepted model. That can be a viva question. So bilayered phospholipid, integral proteins, then peripheral proteins, and then we have lipoproteins and carbohydrates. So another model which is called unit membrane model, that is under electron microscope we can actually see 3 layers. So which 3 layers? That is, phospholipids are sandwiched between the outer protein layer and inner protein layer. So that is the unit membrane model, but most accepted is fluid mosaic model, that it is a mosaic of so many components. Then it lacks sterol, but exception is mycoplasma, where sterols are present in plasma membrane, but sterol-like molecule, that is hopanoids, are present in the plasma membrane. So what are the functions of plasma membrane? It is a semi-permeable membrane. It allows some materials to pass through. It has specific enzymes within it which are called permeases. So it acts as a transport system. Certain nutrients can be taken in and waste material can be excreted. So as so many metabolites pass through, so it is called as site for metabolic processes. So those are the functions. Now let's talk about the cytoplasm. Cytoplasm is a colloidal system, that is, it is made up of solutes in a solution. So inorganic and organic solutes, like in the watery solution, water may comprise about 70% of the cytoplasm. Protoplasmic streaming means the internal movement of this cytoplasm. Protoplasm is absent in bacteria, whereas it is present in eukaryotes. And then the membrane bound organelles like Golgi apparatus and mitochondria are also absent in this cytoplasm of bacteria, whereas they are present in eukaryotes. So what is present in this cytoplasm of bacteria? Ribosomes, inclusions, and mesosomes. So let's see what are ribosomes. They are the sites of protein synthesis, and they are in RNA. They also get integrated in mRNA. Their size is smaller than eukaryotic cells. They have 70s size. Now what about the inclusions? Inclusions which can be seen are of different types, like volutin, there can be their polysaccharide and lipids. Now why these inclusions are formed inside this cytoplasm? So they are called as energy reserves, that is, they are the storage product of the bacterial cell. So when are they seen? Whenever there will be nutritional deficiency, their storage product will be formed. But when the nutrients are supplied, so these inclusions will disappear. So how do we see these inclusions? Volutin are granules which are seen in the diphtheria bacilli. So they can be seen by a special stain which is called the Albert stain, as we discussed in the staining methods. So these are the metachromatic granules and they can be seen with the Albert staining in diphtheria bacilli. Polysaccharides can be seen with iodine, and lipids can be seen with fat soluble dyes like Sudan black. Then another thing, that is vacuoles can be there. So what are vacuoles? How are they different? So vacuole can be a fluid containing cavity. So they can be a cavity in which the fluid may be present, and they are separated from the cytoplasm by a membrane. So they may have a membrane like this. So this is vacuole and this is inclusion. So those two can be present. Then third is mesosomes. So mesosomes are also called chondroids. What are they? They are the invaginations of the plasma membrane. Plasma membrane may be invaginated towards the cytoplasm in the shape of vesicles like this, or it can be in the shape of tubules, or it can be in the form of lamellae or layers. So those are chondroids or mesosomes. What is their role? They are prominent in gram-positive bacteria. They are sites of bacterial respiration. That means they have some respiratory enzymes. So they are considered analogous to mitochondria of eukaryotes. So what is mitochondria to eukaryotes? That is mesosomes to prokaryotes, that is bacteria. They can also help in cell wall formation and chromosome replication. Now what about nucleoid? So nucleoid, true nucleus is absent in bacteria. So there will be no nucleolus, no nuclear membrane, but genetic material is in the form of nucleoid, which is usually one per cell, which is usually irregular shape, or it can be oval or elongated. So it is made up of single haploid chromosome comprised of supercoiled circular double-stranded DNA. It is coiled, but when it is open, it can be of length up to 1000 micrometers, and then it is not associated with basic protein. So how do we see this nucleoid there? It can be seen under electron microscope or with a special stain which is called Flagella stain, which can be important viva question. So plasmids or episomes, that can be another important short note or viva question. So what are they? So DNA can sometimes lie outside this nucleoid in the cytoplasm freely. So as we can see here, so this is called plasmid. So this is extranuclear DNA that is outside the nucleoid region. They are present in cytoplasm. So what is their role? They are carriers of genetic information. They can carry genetic information during the division of the cell. So that is binary fission. So they can carry the genetic information from one bacterium to another through conjugation, through bacteriophages. Now they plasmids can give special characteristics to bacterial cell like toxicity and drug resistance. Present, they are not essential for life. When they are present in the bacteria, they can give drug resistance to bacteria against many drugs. So they can have survival advantage, these bacteria. So we have discussed cell wall, plasma membrane, cytoplasm inclusions, and nucleoid, and the remaining, that is cell wall appendages, atypical forms, and bacterial spores, we'll discuss in our next video. So let's check what have you learned. So first question, where do you see this? Whether in gram positive or gram negative, where you find thicker peptidoglycan, teichoic acid, outer membrane, lipopolysaccharides, and endotoxins. You have to choose one. Then second is not true for bacteria. They have cell wall, they have cell membrane, they have either DNA DNA or RNA, and then they are unicellular. So out of these, which one is wrong? So plasmids are extranuclear DNA. They are also called episomes. They are carriers for genetic information, or all of the above. And finally, is not what is true about bacterial inclusions. They are intracytoplasmic, they are storage sites, they are seen in nutritional deficiencies, or all of the above. So that is all for this video. If you really enjoyed the video, do tap on the like button and share this video with your friends. Keep watching, keep learning, keep smiling. See you in the next video soon. Till then, take care. Bye bye.